Alright everybody, welcome to my Exam 2 Essay 1 video. First off, we're going to start talking about muscle contraction. We're going to start in the skeletal muscle and then we'll move over to the smooth muscle really quick. So to start off, let's draw what's called the neuromuscular junction. So right there, that's going to be our presynaptic terminal. And this is going to be our sarcolemma, which is essentially the plasma membrane of our sarcomere, or our muscle cell. And let's see here, let me draw a few things. Right here, this is going to be a voltage calcium channel, okay? And we're going to have some vesicles here, some transport vesicles in our presynaptic terminal that are going to be filled with acetylcholine that we heard of before. Now on our postsynaptic terminal, in this case the sarcolemma, we're going to have some cholinergic receptors. In this case, these are nicotinic 1 receptors, so N1 receptors. And then here we're also going to have these are ligand-gated cation channels, and the ligand is going to be on our N1 receptors here, okay? I think that's all we need. Okay, so to start off, first thing, also in our presynaptic terminal, we're going to have what's called snare proteins. They're going to be on our transport vesicles and on that inner membrane of our presynaptic terminal. Okay, so the first thing that's going to happen is we're going to have an action potential. It's going to come down our presynaptic terminal and it's going to hit this voltage gated calcium channel. What that's going to do is it's going to open and it's going to allow calcium ions to rush into our presynaptic terminal. And what this calcium does is it's going to bind to our snare proteins. And when it binds to our snare proteins, they're going to kind of twist together, kind of pull like in closer together. So it's going to pull these transport vesicles towards our synaptic cleft. So right here's the synaptic cleft. And so essentially what happens is via exocytosis, our presynaptic terminal is going to kick out those vesicles of acetylcholine and it's going to diffuse into our synaptic cleft. And eventually those little acetylcholines are going to bind to our N1 receptors. And when those ligands bind to our N1 receptors, it's going to open up our cation channels and allow lots of sodium and calcium and whatnot, but lots of positive charges to flow into our sarcomere through the sarcolemma, okay? Now also in our synaptic cleft, we're going to have an enzyme. And this enzyme is called acetylcholine esterase. And what that enzyme does is it goes through and just eats up this acetylcholine and breaks it into the cholyl groups and the acetyl groups and kind of cycles it through so it can be recycled into more acetylcholine later. We're not going to get into that too much, but acetylcholine esterase, so it goes through, wipes out acetylcholine so you don't have too many muscle contractions, because that could lead to spastic paralysis, which we wouldn't want. So once these positive charges come in, it's going to depolarize this membrane. And so it's going to travel down, it's going to continue to depolarize. Now right here is kind of where it changes. So this is our sarcolemma. It's going to dip down. And this thing right here we would call a T-tubule. Now that's one difference between skeletal muscle and smooth muscles. Our smooth muscle does not have these T-tubules, but our skeletal muscle does. And so essentially our smooth muscle, it has to use G proteins and other ligand channels, voltage gated calcium channels, to get calcium into its cytoplasm to innervate a muscle contraction. I won't get into that too much. We're going to focus on skeletal muscle right now. And so on our skeletal muscle here, so let's draw this too. So right here, this is going to be our sarcolemmic reticulum, or SR, and inside that we have a lot of calcium ions, okay? And we need these calcium ions to start a muscle contraction, okay? And so on our SR, there is a protein called ryanodine. So this is ryanodine, okay? And attached to ryanodine is a DHP receptor. And this is essentially a voltage-gated calcium channel, but we don't use it to bring calcium in. We could, because there are high concentrations of calcium out here, but we're not going to do that, and we'll show you why. So essentially, go back to our purple marker here, our action potential is going to flow down, it's going to hit that DHP receptor. And then what that does is it's going to open that, but it's going to change the configuration of our ryanodine protein and essentially open it up. Okay, and what that does is now calcium, it would want to drive into this, but our driving force of calcium in here coming out far outweighs the calcium's driving out force out here to come in. So what we're going to see is calcium in our SR going to come and it's going to flow out into our cytoplasm, okay? Now we're going to pause there and we're going to kind of show what happens right here at this step with calcium. And so to go through, let's move over here, see how well I can do this so everybody can see it here. So on our, this is what's called a sarcomere. Okay, what a sarcomere is, it's a different unit, or essentially just one motor, not a motor unit, but just a unit in our muscle cells that we're going to come closer together and kind of overlap, essentially. And that's what causes our muscle fibers to shorten and lengthen, so to contract and relax. And so this right here, this is called our Z disc. So a sarcomere is essentially one Z disc to the next Z disc. And this protein, this filament, that's called nebulin, and then this other one is called titin. And so on our nebulin protein, we're going to have all these G-actin subunits. We're going to kind of bind to it. And all these G-actin subunits together, they make an F-actin. And all of our G-actin subunits, they have a myosin binding site. And we'll show what myosin does here in a minute. But right now, covering those myosin binding sites, there's another protein called tropomyosin. And tropomyosin just covers those myosin binding units. And attached to our tropomyosin and our actin, we're going to have kind of this little like three-balled protein called troponin. And troponin has three different subunits. It has a C subunit here, an I subunit, and a T subunit. Okay. Now our C subunit has 4 different binding sites for calcium. So it has high affinity to calcium, but on these binding sites, 2 of them are high affinity calcium. So it's always going to have some calcium attached to it. And these other 2 need the calcium here to bind to it. Okay, and essentially this subunit, it just alters the positioning of this protein. And our T subunit is attached to the tropomyosin. It has a high affinity for tropomyosin. And our I subunit has a high affinity for actin. So it's going to be bound to our actin there. And so when our calcium here comes in and it binds to that C subunit, our troponin, it's going to alter that position and pull this tropomyosin out of the way to expose our myosin binding sites on our actin. Now our myosin, so this is a thin filament also in this whole thing, we kind of just refer to it as actin, but our myosin is going to look something like this. It's going to have all these different myosin heads that want to bind to the myosin binding sites, but they can't because tropomyosin is in the way. So once calcium binds to our C subunit on troponin and pulls that tropomyosin away, these myosin heads are able to come up and bind to that, okay? Now we're going to show what's called the cross-bridge cycle, which essentially is what kind of develops our muscle contraction. So right here, here's our Z disc, and then this is just going to be our myosin, this is going to be our actin. And so right now, for step one, our myosin head is going to come up and it's going to have an ADP attached to it. And one thing to remember about our myosin heads is an ATPase, so it's going to hydrolyze ATP. And it's also going to have an inorganic phosphate right there, okay? So now, we're going to come over here, we're going to introduce calcium. So it's going to bind to our actin. And what the calcium does is it binds and it exposes that tropomyosin. And now our myosin is able to bind to our actin, and it's going to release some energy and heat out of it, but it's also going to release some energy that's going to go in and break that phosphate off. So now we go to our next step here. So now our phosphate is going to come and break off here. And the energy from that is going to cause our myosin head to bam, to snap that way, okay? And that just moves our whole actin, it moves our whole sarcomere that way. So I have some movement here, and this is what's called power stroke, okay? But now we need to reset it because it's kind of stuck there, but we want it to keep going, to keep power stroking, to keep causing the muscle to contract. So then what we need to do, let's see how we're going to draw this here. Now let's just do this. Let's do this. So right here in this step, we're going to bring in some ATP. It's going to come in. It's going to bind to the head, and when it does that, it unbinds it from the actin. But because this is an ATPase, I want to reset it to this position. It's going to hydrolyze the ATP and reset it into this, okay? So that is what's called our cross-bridge formation for skeletal muscle. Now, as we know, with skeletal muscle and smooth muscle, there's some differences. We already showed that our skeletal muscle is going to have T-tubules, smooth muscle is not. Smooth muscle is also, for the most part, involuntary, and skeletal muscle is mostly voluntary. Now, it's really beneficial as far as smooth muscle goes with regulating blood vessels, our sphincters, digestive system. So it's really nice to have that as involuntary. But also, because of those differences, it kind of has a different cross-bridge cycle. So kind of shows some more differences. So up to this point, it's all the same with smooth muscle, other than smooth muscle does not have a T-tubule. So rather than have this T-tubule and ryanodine, there's going to be like G proteins, phosphodiesterase C, and some other channels. Now, if the calcium in our smooth muscle comes from the SR, that's going to cause what's called a phasic contraction, or phasic smooth muscle. What is that too? It has kind of a different cross-bridge formation. Okay, so let me pause really quick and get everything drawn out here. Okay, perfect. So we just cut out a few seconds there just to make sure we're not using too much time on this video. But now back to our phasic contraction. Another thing to remember about smooth muscle is it has no troponin. Okay, so no troponins coming in, which means calcium is not really binding to anything, but we still need calcium very much in this. So calcium is going to come in And then also an enzyme called myosin light chain kinase is going to come in. And what that does is it adds a phosphate to our myosin head and then ATP. Now all that ATP and calcium is going to be able to push the tropomyosin out of the way and allows it to bind. And also this kinase enzyme, MLCK, is going to add another phosphate to our myosin head. So now we have two phosphates there. And kind of the same thing from it binding. It's going to lose some energy, and some of that's going to go into this other phosphate. So then we're going to kick that phosphate out. We're going to add it. We're going to kick out this ADP that was on here, and then that's going to cause our power stroke. So it's going to contract again, still with this second phosphate. And it's going to go in. We're going to add our ATP just like last time, which is going to break that bond. But now what's different here, other than the second phosphate, is with that second phosphate, it can just go skip and just go right to this next stage. So it can kind of just reset, get another reset, doesn't need to add this phosphate, and it can just keep going, just keep cranking the contractions out. But if we want to end the contraction, we want to relax the muscle, then we need this myosin light chain phosphatase, which is going to remove this phosphate, kick it off, and reset our myosin head with the ATP. It's going to hydrolyze still to ADP and phosphate. Okay. Now, like we said, there's phasic and then there's tonic contractions with smooth muscle. And so the difference with our tonic, we get rid of that here. That. Okay. Is now with tonic, we're still going to be up to this point is going to be the same. But then what's going to happen is we're going to kick off that phosphate and our ADP. But then also our MLCP is going to come in early. And so it's going to cut this phosphate off. So it's still going to go through the movement. We're still going to add ATP, but ATP is going to have a hard time binding now. It's going to be kind of locked up. And so what that causes is this is still going to be bound here. So it's going to be bound there, and this is what's called now our latch state. Okay. So now it's locked in there, and it's going to stay that way. It's going to stay contracted until finally ATP is able to finally bind and break it and then reset it again. This is really helpful for, say, your anal sphincter, so you don't poop your pants, so your sphincter will stay contracted. You don't have to worry about pooping your pants, which is always nice, okay? Which is something that plastic or our phasic smooth won't be able to do. Our phasic would be really good for, say, maybe our digestive system. We're constantly needing to just move stuff through, so it's just going to keep contracting and it won't have to use up as much ATP here. Okay, so that's it for our skeletal and smooth muscle contractions. I want to pause the video again, and we're going to move on to energy sources for contraction and also reflexes. Okay, perfect. Now, starting with our energy sources for our muscle contraction. Get my notes here so I know what to say here. So first one is going to be our, as far as ATP. ATP is the main energy source for our muscle contractions. And so our first is going to be our cytoplasmic storage. Okay. Now, this is nice because it's just right there. It's just floating around in the cytoplasm. Our muscle cells can just hurry, grab it, and use it. But the downside is it only lasts about 1 to 5 seconds. So very short-lived contraction from there. But things like our eyes use it a lot, where it's doing those rapid, quick, short contractions. So it is still kind of helpful, okay? But this would be like when you're going through a day and you just need something super quick to keep you going. That's like our granola bar. Just something small, get you going for a little short amount of time. Well, if we need something bigger, like maybe a sandwich, then we would need something called creatine phosphatase. Okay. Now what our creatine phosphatase does is it essentially finds all these ADPs kind of floating around, and it's going to bind to it. So it's going to add a phosphate, or it's going to take this phosphate and add it to ADP to make some ATP. So then it's kind of scounging up, it's kind of picking up the leftovers in our cytoplasmic stores, recycling it. But this, it only lasts maybe 10 seconds. So still very short-lived energy source. But if you're needing to, I guess, contract just a little bit longer, it's a good source. Just a nice rapid source as well. But say we need to run a sprint, so before our sprint we're gonna want to eat like a burger and fries really quick, get some carbs in, some energy. Then we're gonna use what's called glycolysis. Now we've all learned about glycolysis. That's when we break down a glucose molecule and eventually break it down into ATP. And then we can use that ATP in our muscle contractions. Glycolysis lasts about 1 minute as far as contraction. So it's very good to say if you're going to run a full-on sprint, glycolysis is good. It's a quick energy and it lasts a minute. But say now we want to go backpacking and we're going to have to be standing somewhere for hours on end. We're going to need something a little different. These three things, the reason they can't produce such a powerful contraction is because they're anaerobic. They don't use oxygen. So if we want it to last forever, then we would need this last source, which is our aerobic respiration. That's our long-term way to make ATP and produce it using oxygen. You might have heard of like electron transport chain. And this we can just last hours with the ATP we get from aerobic respiration. So very, very, very good source. And again, this is aerobic. It's using our oxygen, okay? And these are anaerobic. So they all play their part. Our muscles will kind of go through everything. So we'll use this first. If it needs more, go down, and as the list goes on, okay? But our aerobic respiration is going to give us the most ATP, which is going to allow for more rapid, stronger muscle contractions, okay? Now, moving on to our reflexes here. We'll go to this side. This is if we say took a muscle fiber or unit and just pulled out of our leg. I'm going to kind of zoom in on it a little bit. We'd see that we have all these fibers running down it. So these are our tendons here, but we'd see these fibers. These are called extrafusal fibers. But then also within it, we're going to have this little purple thing. It won't actually be purple, but just for this sake. And this is going to be our muscle spindle apparatus. Okay, so that's what that is. And now we have different neurons kind of controlling it. So on our normal motor neuron or motor unit, we're going to have our alpha motor neuron. And then for our muscle fiber or our muscle spindle, we're going to have a gamma motor neuron. And then our muscle spindle, it's also going to have some sensory neurons coming out of that. And so if you break it down, we take our muscle spindle apparatus out, and we're going to zoom into that a little bit more, okay? So this kind of fiber that has our nuclei kind of pushed to the side, it's very sandwiched together. That is our dynamic, let me look at my notes really fast here. Our chain fiber, I believe. Yep. Okay, back on track here. So right here, this is going to be our chain fiber okay? And our chain fiber is going to be innervated by a static gamma neuron. And this other one that's got kind of a bulge to it, this is going to be our bag fiber, and it's innervated by a dynamic motor neuron, okay? And we also have sensory neurons in both of these. So our chain fiber, that's going to be our 2A sensory neuron. And then our bag fiber, this is going to be our 1A sensory neuron, okay? We'll kind of break this down. So how our reflexes work is essentially this muscle spindle, it kind of wants to stay at the same length. It doesn't really like to be lengthened. And so say if we're doing our knee jerk test, we push on that patellar tendon, so push on that, it's going to lengthen our muscle spindle, but it doesn't want to do that. So it's going to send a reflex to hurry and contract our quad so it doesn't have to be lengthened as much. And so what's essentially going to happen here is it's going to get pulled and stretched, and as it's getting stretched, this 1A fiber is going to be going crazy as it's getting stretched, as there's movement, and then right as it's stretched to the max, that's when our 2A sensory neuron is going to fire off. When it's all the way stretched, that's 2A, 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 okay? So essentially what's happening at like the spinal cord level, if I remember how to draw this here. Something like that. So this is our dorsal end and this is our ventral end. So essentially what's going to happen is that nerve that's going to come in here, and then it's going to come in again and it's going to synapse with one motor neuron to contract our quad. So it's going to tell our quad to contract, but because we're contracting our quad, we need to relax our hamstrings. But we can't just do that from the same synapse. It's going to kind of branch, synapse with another IPSP neuron, which is going to cause our hamstrings to relax a little bit. Okay? So that's our stretch reflex. It's all the muscle spindle and how it gets lengthened and how it's able to kind of maintain that length. Okay? Now, going back to this, on our tendons here, we're going to have something in there, and this is called our Golgi organ, I believe. Golgi tendon. Anyway, the Golgi is what we're going to go with. Okay. So what that does is it prevents injury. Basically, all these reflexes are all to prevent injury. So say if we put our hand on a hot stove, the nociceptors in our skin is going to tell or hurry and tell our spinal cord, like, hey, that's hot, pull it away. We're going to see this. So we're going to have some contraction of one muscle to pull it away, but also some relaxation of the other. Or if we step on a tack, for example, it's going to do the same thing. We're going to contract that leg, but also we're going to need to contract the other leg so we don't fall over. So essentially these reflexes are just to help us out, to protect our bodies. The Golgi, what it does, the Golgi organ, what it does is it's in this tendon on our muscle, and it just measures how much that tendon is being stretched so it doesn't rupture and break, because that would be very, very bad, okay? So essentially it's going to go kind of do something similar to this. So say we're like lifting a lot of really, really like heavy weights. It's going to see that this tendon is being stretched and contracted way too much. So it's going to send a signal to our spinal cord, which is going to say, hey, you need to release relax the biceps and contract the triceps to relieve that tendon, and then you're just going to drop that weight. But sometimes that fails and it does break. Sometimes we're in like positions where we're not able to relax our muscles. We saw with like Aaron Rodgers, he's kind of in a very, I guess, dorsiflexed position with his calves. I'm sure his Golgi was like, hey, you need to relax that, but in that position, he's not really able to relax it, so then it ruptures anyway, or if there's some sort of anticipatory brain function that thinks something big is going to happen, but nothing does, and it'll kind of twist and it'll snap as well. But so essentially the gist of our reflexes and everything like that. Hope you enjoyed the video. Please give me a good grade. Thank you.